Have you ever wondered how your body gets energy from the food you eat? Whether you're running, studying, or even sleeping, your body is constantly performing chemical reactions to keep you alive. This process is called metabolism. In biochemistry, metabolism refers to the set of reactions that break down nutrients to release energy, which is called catabolism, and build essential molecules needed for growth and repair, which is called anabolism. These processes are powered by enzymes and are crucial for everyday functions, from digesting food to healing wounds. Understanding metabolism not only explains how our bodies work, but also helps in addressing real life health issues like obesity, diabetes, and fatigue. Today, the amines group from BS Chemistry 4.1 and 4.2 will help you dive into the fascinating world of metabolism. We'll explore how our cells ingeniously transform the food we eat into usable energy, specifically ATP, through a remarkable process called cellular respiration. We will also uncover the essential accelerating and regulatory role that enzymes play in every single one of these vital biochemical reactions. So, first of all, what exactly is metabolism? Simply put, it is the sum total of all the chemical reactions occurring within an organism. Think of it as the intricate biochemical machinery that keeps us alive. Metabolism can be broadly divided into two main categories. First, we have anabolism. These are the building up reactions. Anabolic pathways consume energy to construct complex molecules into simpler ones, things like synthesizing proteins from amino acids or building DNA. Second, we have catabolism. These are the breaking down reactions. Catabolic pathways release energy by breaking down complex molecules into simpler ones, for instance, breaking down glucose to release energy. Together, these anabolic and catabolic reactions enable our cells to grow, to reproduce, and to maintain their structures and respond dynamically to the changes in their environment. Let's proceed to the functions of metabolism. Metabolism serves three critical functions that are fundamental to life. Firstly, it is how our bodies convert the food we eat into usable energy, primarily in the form of ATP. ATP is the cellular currency that powers everything we do, from thinking to running. Second, metabolism is responsible for building and maintaining all of our cellular structures. This includes synthesizing of all the proteins, the lipids, carbohydrates, and nucleic acids that make up our cells and tissues. And thirdly, it plays a vital role in eliminating metabolic waste products such as ammonia, which, if allowed to accumulate, would become highly toxic to our systems. Proceeding to the catabolism pathways. So let's dive a bit deeper into catabolism. As I have discussed, these pathways are all about breaking down complex molecules and in doing so, releasing energy. A classic example of catabolism is the digestion of proteins. When we eat protein, our digestive system hydrolyzes these large molecules into their individual building blocks, which are the amino acids. But what happens to excess amino acids that aren't needed for building new proteins? So the amino acids undergo a process called deamination, which produces ammonia, which is a highly toxic compound. Fortunately, our bodies have a clever way to deal with this. In humans, ammonia is converted into a less toxic form, which is urea, through a series of reactions known as the urea cycle. As you can see on the slide, the mechanism involves several steps, starting with ammonia and carbon dioxide combining to form carbamoyl phosphate and eventually leads to the production of urea, which is then secreted from the body. Now let's switch gears to anabolism. Anabolism is the building up processes that require energy typically supplied by the adenosine triphosphate or the ATP. A prime example of this is the protein synthesis, where individual amino acids are meticulously joined together to form long protein chains. This joining happens via a peptide bond, which is formed in a dehydration reaction, which means a molecule of water is removed. And this process is catalyzed by ribosomes within our cells. It's not just in animals. Plants also use similar dehydration mechanisms. For instance, they build complex protective molecules like condensed tannins from simpler anthocyanins units. Proceeding to the ATP, or the adenosine triphosphate, central to all metabolic processes is this, the ATP, or the adenosine triphosphate. Think of ATP as the universal energy currency of the cell. Its structure is the key. It stores a significant amount of energy in the bonds between its three phosphate groups. When that outermost phosphate bond is broken, a burst of energy is released. This process is called the ATP hydrolysis. As you can see, when ATP reacts with water, it breaks down into ADP, or the adenosine diphosphate, and an inorganic phosphate, releasing approximately 7.3 kilocalories of energy per mole. This released energy is then immediately used to power virtually all cellular work, from muscle contraction to active transport across cell membranes.
Building on our understanding of ATP as the cell's energy currency, let's now turn our attention to the main way most living organisms produces this vital molecule, through the remarkable process of cellular respiration. So, cellular respiration is not just any process. It is a quintessential catabolic pathway. Remember, catabolism means breaking down molecules to release energy. In this case, our primary fuel source, glucose, which we obtain from the food we eat, is systematically dismantled step by step to release a substantial amount of chemical energy, which is then captured and stored in the bonds of ATP. The overall chemical equation for cellular respiration, which you see on the slide, summarizes the grand transformation. So, in the reaction, one molecule of glucose combines with 6 molecules of oxygen to yield 6 molecules of carbon dioxide, 6 molecules of water, and critically a significant net amount of ATP. Beyond simply showing the inputs and outputs, it's crucial to understand that cellular respiration is fundamentally a redox process. So, in this reaction, glucose is oxidized. This means it loses electrons, and as glucose is broken down, the electrons are gradually stripped away. So, oxygen is reduced. Conversely, oxygen gains the electrons. So, oxygen is the final electron acceptor in this intricate process. This movement of electron from glucose to oxygen is what ultimately drives the energy capture for ATP synthesis, making it a highly efficient energy generating pathway. And to further our discussion, the four stages of cellular respiration, namely glycolysis, pyruvate oxidation, Krebs cycle, and electron transport chain, will be discussed.
Cellular respiration is more than just a chemical reaction. It is a finely tuned series of steps that ensures our body get the energy they need. Let's begin with glycolysis. Glycolysis takes place in the cytoplasm of the cell, outside the mitochondria. Here, a single molecule of glucose, a simple sugar that we get from the food we eat, is split into two molecules of pyruvate. This process doesn't require oxygen. which means it can happen in both aerobic and anaerobic conditions. The process begins when the cell invests a little energy by using ATP to add phosphate groups to glucose, which will help activate and commit the glucose to the pathway. Next, the six carbon sugar is split into two 3 carbon molecules by the enzyme aldolase. In the later steps, energy is harvested as these 3 carbon molecules are converted into pyruvate, producing ATP directly through substrate level phosphorylation. During glycolysis, the cell invests a little energy to start the process, but the payoff is greater. For each glucose, the cell gains a net total of 2 ATP, 2 NADH, and 2 pyruvate molecules that will move on to the next stage. Once the glycolysis is complete, the 2 pyruvate molecules move into the mitochondria, the cell's powerhouse. Here, each pyruvate is converted into a molecule called acetyl-CoA. This conversion is not just a simple handoff. It is a critical point that links the process of glycolysis to the more complex energy extraction in the next stage. During this step, each pyruvate loses a carbon atom, which is released as carbon dioxide, a waste product that we eventually exhale. The process also generates NADH, another energy carrier. Now we enter the Krebs cycle, also known as the citric acid cycle, which takes place in the mitochondrial matrix. This cycle is a series of chemical reactions that further break down acetyl-CoA. Each turn of the cycle generates more high energy molecules, which are 3 NADH, 1 FADH2, and 1 ATP per cycle, and releases 2 more molecules of carbon dioxide as waste. Since each glucose molecule produces 2 acetyl-CoA, the cycle spins twice for each glucose. The main function of the Krebs cycle is to harvest electrons by transferring them to NAD plus and FAD, forming NADH and FADH2. These molecules are like fully charged batteries that will deliver their energy to the final stage. Now let's move to the electron transfer chain, or ATC. This stage is where most of the ATP is made. The ATC is located in the inner membrane of the mitochondria. Here, the NADH and FADH2 produced in previous steps donate their high energy electrons to a series of protein complexes. As electrons move down the chain, their energy is used to pump protons across the membrane, creating a gradient. This gradient is used by the enzyme ATP synthase to generate ATP, about 26 to 28 molecules per glucose, far more than any previous stages. At the end of the chain, oxygen acts as the final electron acceptor, combining with electrons and protons to form water. Without oxygen, the entire process would back up and stop. The ETC is the ultimate energy payoff, converting the stored energy from earlier stages into a usable form for the cell. So, to wrap up, cellular respiration is a multi-step process that allows cells to efficiently convert the energy in glucose into ATP. Um, each stage, which are glycolysis, pyruvate oxidation, the Krebs cycle, and the electron transport chain, plays a unique and vital role in cellular respiration.
Moving on to understanding the overall process by quantifying the energy produced, we have energy counting. Energy counting is the process of measuring the amount of ATP, NADH, and FADH2 produced or used during cellular respiration. This quantitative approach is essential for several reasons. First, it allows us to assess the efficiency with which cells convert nutrients such as glucose into usable energy. By precisely tracking these energy carriers, we can determine how effectively the cell maximizes the energy output from each glucose molecule. Second, energy counting enables a comparison between different metabolic pathways. For instance, we can compare the energy yield of aerobic respiration, which uses oxygen, with that of anaerobic processes, which do not. Third, the measurement helps explain adaptation at the cellular level. Some cell types, such as red blood cells, rely exclusively on glycolysis, while most others utilize the full aerobic pathway. Energy counting provides a rationale for these adaptations based on the energy demands and constraints of the cell. The procedure for energy counting is systematic and follows a clear sequence. First, we identify the number of NADH and FADH2 molecules generated per glucose molecule at each stage of respiration. These electron carriers are produced in glycolysis, pyruvate oxidation, and the Krebs cycle. Second, we can determine the ATP yield per NADH and FADH2 molecule as they pass through the electron transport chain. In most eukaryotic cells, each NADH typically yields approximately 2.5 ATP, and each values may vary slightly depending on the cell type and the specific shuttle mechanisms involved. Third, we can add the direct ATP produced by substrate level phosphorylation in glycolysis and the Krebs cycle. Finally, we sum these values to obtain the total ATP yield per glucose molecule. This step-by-step ensures that we capture all sources of ATP production and provides a comprehensive view of cellular energy metabolism. A critical aspect of energy counting is distinguishing the theoretical maximum and the actual yield of ATP per glucose molecule. The theoretical maximum is often cited as up to 38 ATP per glucose molecule. This value is based on ideal conditions where every NADH and FADH2 is converted to ATP and no energy is lost. However, in actual eukaryotic cells, the yield is typically about 29 to 30 ATP per glucose molecule. This discrepancy arises due to several factors. First, energy is expanded to transport molecules such as pyruvate into the mitochondria. Second, protein leakage across mitochondrial membrane reduces the efficiency of oxidative phosphorylation, leading to a loss of potential ATP. Third, there is a variability in the exact number of protons required to synthesize each ATP molecule, further influencing your final yield. Understanding these differences is crucial for appreciating the real world constraints on cellular energy and production and the importance of accurate energy counting in biochemical studies.
We've been talking about all these complex reactions: glycolysis, the Krebs cycle, oxidative phosphorylation, but how do they happen so quickly and efficiently inside our bodies? The answer lies with enzymes. Enzymes are incredible biological catalysts. That means they help speed up biochemical reactions without being used up in the process. Their most important role is lowering what is called the activation energy. It is the energy barrier that a reaction needs to overcome before it can proceed. Think of activation energy as a hurdle. Without enzymes, the molecules trying to react have to jump really high to get over that hurdle. But enzymes provide a sort of a ramp, like an alternative pathway that makes it easier for the reaction to take place. This is why reactions that might take hours or even days in a test tube can happen in just seconds in our cells. And it also happens in a body temperature. Enzymes do this by binding to specific molecules called substrates. They hold these substrates in the perfect position to make a reaction happen more easily. This creates what's called a transition state, which is like the halfway point between reactants and the product. Once the reaction is completed, the enzymes releases the final product and is free to catalyze another reaction. So, in short, enzymes make metabolism possible. Without them, life as we know it wouldn't be fast or efficient enough for us to survive.
The study of how enzymes function and how reaction rates are influenced by various factors is called enzyme kinetics. One of the most fundamental relationships we examine is between the reaction rate and the substrate concentration shown in the graph in this slide. As we begin to increase the substrate concentration, the reaction rate also increases. This is because more substrate molecules are available to bind with the enzyme's active sites, resulting in more product formation. However, this increase doesn't continue indefinitely. Eventually, the enzymes become saturated and every active site is occupied, leading to the reaction reaching the maximum rate known as the Vmax. At this point, even if we add more substrate, the rate of reaction doesn't significantly change because the enzymes are working at full capacity. The substrate concentration at which the reaction rate is half of Vmax is called the Michaelis constant or Km. Km gives us insight into the enzyme's affinity for the substrate, and a lower Km means higher affinity since it takes less substrate to reach half maximal velocity. In connection with this, let's now move forward to the next slide, which is the Michaelis-Menten kinetics. To quantitatively describe the relationship between substrate concentration and reaction rate, we use the Michaelis-Menten kinetics model. This model introduces two crucial parameters. First, we have the Vmax, or the maximum velocity, which is the fastest rate an enzyme catalyzed reaction can reach when the enzyme is fully saturated with the substrate. At this point, all enzyme active sites are constantly occupied. Second, we have Km, or the Michaelis constant. It is the substrate concentration at which the reaction rate is half of Vmax. It's a key indicator of an enzyme's affinity for its substrate. Although Km means high affinity, the enzyme reaches half maximal velocity even at low substrate levels. A high Km indicates the opposite. As shown in the graph, this relationship follows a hyperbolic curve. The Michaelis-Menten equation mathematically models this, allowing us to predict how the reaction rate changes based on the substrate concentration and the enzyme's characteristics.
Next, I'll be talking about a crucial aspect of cellular metabolism, the enzyme regulation. In every cell, metabolic processes are carried out by complex pathways composed of multiple enzymes. These enzymes act in a sequence, where the product of one reaction becomes the substrate for the next. While most enzymes follow basic kinetic rules, certain enzymes in these pathways play a regulatory role, meaning they control the overall pace of the entire sequence. These regulatory enzymes are especially important because they allow the cell to adjust its metabolism according to its current needs, whether that's generating more energy, building molecules for growth, or repairing damage. The following are the four main mechanisms. First, the allosteric regulation. Second, the feedback inhibition. Third is the reversible covalent modification. And lastly, the regulatory proteins and proteolytic cleavage. Allosteric regulation is reversible and works through the non-covalent binding of molecules called allosteric modulators. These bind to an allosteric site, which is different from the enzyme active site. This binding causes a conformational change that either activates or inhibits the enzyme. For visual example, on the left side, we see how an inhibitor binds to the allosteric site and distorts the active site so the substrate can no longer bind. And this is allosteric inhibition. On the right side, we see allosteric activation, where an activator binds and stabilizes the active form of the enzyme, making the active site functional. Second, for feedback inhibition, in feedback inhibition, the end product of metabolic pathway binds to a regulatory enzyme early in the sequence to shut the pathway down when enough product has accumulated. For example, the threonine to isoleucine pathway in bacteria. Threonine is converted to isoleucine in five-step enzymatic pathway. When isoleucine levels rise, it binds to the first enzyme, then the threonine dehydratase at an allosteric site and inhibits it. This is called heterotropic allosteric inhibition, where the inhibitor is not the substrate. Once isoleucine drops, the inhibition lifts and the pathway resumes. This ensures resources aren't wasted and the cell maintains balance. And for the third mechanism, the reversible covalent modification. In this mechanism, enzymes are modified by the addition or removal of chemical groups, usually to specific amino acids. One of the most important types is phosphorylation. In this example, the enzyme starts as an unmodified protein. A kinase uses ATP to phosphorylate it, forming an active phosphoprotein. Later, a phosphatase removes the phosphate, returning the enzyme to its original state. This regulation is rapid, reversible, and essential in processes like signal transduction. And lastly, regulatory proteins and proteolytic cleavage. Some enzymes are regulated by binding to regulatory proteins that activate or inhibit them. Others are controlled by proteolytic cleavage, a permanent cut that activates the enzyme. In this example, the enzyme starts as an inactive precursor, also called zymogen. A proteolytic cleavage removes a specific segment, irreversibly activating the enzyme. This is commonly seen in enzymes like digestive proteases or blood clotting factors, where irreversibility is crucial for function. To conclude, the enzyme regulation is essential for metabolic efficiency and adaptability. Cells use allosteric modulators, feedback inhibition, phosphorylation, or proteolytic cleavage to fine-tune enzyme activity based on their needs. Each regulatory mechanism allows the cell to respond quickly and accurately to changes in its environment or internal signals.